Modern clinical trials evaluating multi-organ disease burdens operate at a massive scale. In critical care, cardiology, and nephrology, measuring a drug's true efficacy requires capturing a multitude of interconnected adverse events over long periods of time. Historically, the standard analytical engine for these trials has been the time to first event, or TTFE, composite endpoint. This framework, powered by the Cox proportional hazards model, measures the exact time elapsed from randomization to the very first occurrence of any adverse event defined within the trial's parameters. The mathematical flaw embedded within the TTFE framework is its egalitarian treatment of clinical events. The model assigns identical statistical weight to a transient morbidity, like a localized infection, and an irreversible catastrophic outcome, like mortality. On this timeline, patient A is hospitalized on day 30 and suffers fatal cardiac arrest on day 100. Patient B avoids hospitalization but dies on day 31. The TTFE algorithm flags patient A's day 30 hospitalization and stops evaluating, remaining mathematically blinded to their death. The algorithm actively penalizes patient A for the early minor event. Conversely, patient B is recorded as having an event later, despite dying 69 days earlier. The model rewards the active treatment arm for delaying a hospitalization, while effectively ignoring survival duration. Because of this dynamic, a highly effective life-saving therapy can yield a neutral or even negative trial result. By removing the competing risk of death, the drug leaves patients alive longer to accumulate lesser morbidities, which can paradoxically drive up the event rate and mask the mortality benefit. To resolve this truncation bias, Stuart Pocock and colleagues introduced a different statistical framework in 2012, the win ratio methodology. This method abandons the evaluation of aggregate survival curves entirely. It relies on a mechanism of exhaustive, generalized pairwise comparisons to isolate true therapeutic benefit. Instead of treating all outcomes equally, the algorithm structures the constituent elements of a composite endpoint into a rigid, predefined clinical hierarchy. Irreversible events, like cardiovascular mortality, are placed at the absolute apex of statistical priority. Lesser morbidities are then tiered below in descending order of clinical severity. By forcing a strict hierarchy, the win ratio circumvents the need to assign arbitrary numerical weights, avoiding the interpretive challenges of the TTFE model. This structure locks in the mortality benefit. Non-fatal morbidities are only evaluated if patients share identical outcomes regarding mortality, preventing lesser events from statistically diluting the survival signal. The win ratio aligns the mathematical test statistic with actual patient-centered clinical priorities, ensuring that a statistical victory accurately reflects a genuine, meaningful extension of life or prevention of severe disease. Executing a generalized pair-by-pair comparison requires significant computational infrastructure. It evaluates data at a granular, pair-by-pair level across the entire trial cohort. Early iterations attempted to match specific patients across arms based on baseline risk scores. Because perfect clinical matches result in discarded data, the unmatched win ratio approach is now standard. The unmatched approach dictates exhaustive combinatorial logic. Every single patient in the treatment arm is systematically compared with every single patient in the control arm. This array visualizes the resulting matrix. The total universe of pairs is the Cartesian product of the two cohorts, the number of patients in the treatment arm multiplied by the number of patients in the control arm. In a trial enrolling 1,000 patients in the active arm and 1,000 in the control arm, this methodology generates exactly 1 million mutually exclusive patient pairs for evaluation. This exhaustive matrix ensures no patient data is left unutilized. Every individual longitudinal trajectory contributes mathematically to the final statistic. Looking at this structural flowchart, we can trace exactly how the algorithm evaluates those 1 million pairs against the clinical hierarchy. The evaluation begins exclusively at step 1, the apex of the hierarchy, typically cardiovascular death. The algorithm compares the survival time of the treatment patient against the control patient. If the control patient experiences the fatal event earlier in the trial, the treatment patient is declared the winner. The pairwise comparison terminates instantly, and the lower tiers are never accessed. If the comparison cannot be resolved, if both patients survive the entire trial, or neither dies before being censored, the algorithm registers a tie at the highest tier.
Registering a tie at Tier 1 forces the algorithm to cascade down to Tier 2.
This second tier acts as the clinical tiebreaker, evaluating non-fatal morbidities like heart failure hospitalizations to resolve the pair. If the pair remains tied on morbidities, the evaluation drops to the lowest tiers. Here, the mechanics shift from time-to-event survival analysis to direct numerical comparisons of continuous data, like quantitative biomarkers. To prevent statistically trivial differences from driving wins at this level, such as a 1-meter difference in a walk test, modern implementations enforce predefined thresholds of minimal clinical importance. If that margin isn't exceeded, it registers as a definitive tie. Once the algorithm finishes evaluating all pairs, the outcomes are aggregated into 3 mutually exclusive totals: total treatment wins, total control losses, and unresolved ties. The primary test statistic, the win ratio, is a simple relative quotient, total treatment wins divided by total control losses. Notice that the win ratio formula explicitly excludes the tied pairs. If a trial has extremely low event rates, the denominator shrinks, making the ratio highly volatile. To stabilize the results, statisticians use the net benefit and win odds, which reintegrate the ties. The net benefit divides the difference between wins and losses by the entire universe of pairs. Operating on a scale from -1 to +1, it provides an absolute measure of the treatment effect size while accounting for the baseline event rate of the population. A major concern for regulatory committees is whether replacing TTFE with the win ratio sacrifices the statistical power needed to detect a true treatment effect. Simulation models show the opposite in scenarios where survival curves cross, common in aggressive interventions where initial risk is high but long-term benefit is substantial. The log rank test averages these hazards, often neutralizing the observed benefit. By ignoring proportional hazard constraints, the win ratio can yield power gains exceeding 50% in these specific scenarios, detecting differences where traditional models suffer severe analytical breakdown. Despite its power advantages, the win ratio faces challenges when dealing with right-censored longitudinal data. The most rigorous critiques focus on how to define the estimand, the true population-level quantity we are trying to measure. When patients drop out or trials end, data is right-censored. In the win ratio algorithm, the comparison window between any 2 patients is strictly limited to their minimum shared follow-up time. This sliding window can bias the outcome. A 5-year window increases the probability of resolving a pair through mortality. If censoring truncates that to 1 year, the pair likely ties on Tier 1 and cascades to secondary morbidities. Without a strictly specified timeframe, the unadjusted win ratio is an arbitrary weighted average dependent on the random censoring distribution of a specific trial. Because of this underlying randomness, executing accurate cross-trial comparisons becomes mathematically difficult. To resolve this, biostatisticians use the non-parametric tau-restricted win ratio. This approach artificially imposes a fixed, uniform maximum censoring time, tau, for all pairwise comparisons across the entire study. Mechanically restricting the timeframe induces artificial missingness for patients censored prior to tau. To correct this, statisticians apply inverse probability of censoring weighting to ensure the resulting sample remains representative of the intended trial population. While computationally intensive, imposing the tau restriction is necessary to untangle the true underlying treatment effect from the noise of trial-specific censoring. The win ratio also exhibits high statistical sensitivity. A small structural change to the underlying hierarchy can substantially alter the final output. The calculation is mathematically sensitive to the subjective ordering of the clinical endpoints. We can observe this in a trial evaluating an enzyme replacement therapy for late-onset Pompe disease. The trial assessed 2 co-primary domains: physical endurance, tracked via a 6-minute walk test, and respiratory function, tracked via forced vital capacity. Prioritizing the walk test at the top of the hierarchy yielded a robust win ratio of 2.37, but swapping the priority to place respiratory function at the apex caused a substantial shift. The win ratio immediately dropped to 2.02. This variance demonstrates why the win ratio is not interchangeable with a traditional hazard ratio. It is a non-parametric odds metric representing the probability of achieving a better prioritized outcome, not a measure of instantaneous risk.
To prevent post hoc data dredging, the exact sequence of the clinical hierarchy must be exhaustively pre-specified in the statistical analysis plan before any data is ever unblinded.